Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic sexual reproduction in the flowering plants. So this lecture is the continuation of the previous lecture. So in the previous lecture what we were studying about the embryo sac, okay, and then we studied about the pollen pistil interaction, okay. So in that what you have studied how the pollen is compatible or incompatible. Okay, so the pollens, they produce the pollen proteins which are recognized by the receptors present on the stigma, and then if the pollen is recognized, then it forms the pollen tube, and the pollen tube consists of the male gametes which then enter into the embryo sac, okay, through the micropylar end of the embryo sac through one of the synergids. Clear? And we have studied the role of the filiform apparatus also. Clear? Now this lecture is the continuation of the previous lecture, and in this we'll talk about the double fertilization. Okay, so first of all, I'll give you a brief idea about the development of the embryo sac, and then we'll study embryo sac in detail, and then we'll study about the double fertilization. So this double fertilization is seen in the angiosperms. Okay, so let's start this lecture. First of all, let's start with the development of the embryo sac. So development of the embryo sac is also known as megasporogenesis. And embryo sac is known as the female gametophyte. Embryo sac is the female gametophyte and pollen grain is the male gametophyte. Now this embryo sac is present within the ovule. Okay, and ovule is present within the ovary. And ovary is the part of what? It is the part of the pistil. Now first step. What is the first step in the formation of the embryo sac? First is the formation of the megaspore mother cells that are formed at the micropylar region. We have already discussed in the previous lectures. Okay, briefly we have discussed there. First is the formation of the megaspore mother cells. So megaspore mother cells are diploid, means they contain 2 sets of chromosome. Now within the ovule, the diploid megaspore mother cells, they undergo the meiosis. So meiosis always occurs in the diploid cell. Generally it occurs in the diploid cell. To produce 4 haploid megaspore, means a megaspore tetrad is produced. Now meiosis results in the formation of the tetrad, but typically only one functional megaspore survives while the other degenerates. So what does this mean? So there is a megaspore mother cells, okay, that is produced at the micropylar end. That is produced at the micropylar end. Clear? So there is one megaspore mother cell that is diploid and it undergoes meiosis. Meiosis is known as reductional division, and at the end of the meiosis, how many cells are produced? 4 cells are produced. Clear? So a tetrad is produced. Tetrad means 4 cells. Okay, 4 cells are produced. So this is a tetrad. This is known as megaspore tetrad. Now what happens? 3 megaspore degenerates. Okay, out of these 3 will degenerate. Okay, so 3 megaspore degenerate and only 1 megaspore is functional. Alright, this megaspore then forms the embryo sac. Clear? Now let's move on to the next slide. So let's talk about the megaspore development. Now the surviving megaspore, the functional megaspore, we can say that this as the functional megaspore also. These megaspore undergo subsequent divisions often involving 3 mitotic divisions. So it undergoes 3 mitotic divisions. Clear? To form a 7-celled and 8-nucleate structure, which is known as the embryo sac. Clear? So in the previous lectures, what we have studied, initially megaspore divides to form 2 cells. Okay, initially 2 cells are formed. Then from the 2 cells, 4 cells are formed. Okay, and then from the 4 cells, 8 cells are formed. Okay, so these are the 8 cells that are formed, and these, okay, and then what happens? So initially there is no cell wall formation. Initially there is free nuclear divisions. Okay, what are free nuclear divisions? All the nuclei, they keep on dividing, and there is that, and the nuclear division is not followed by the cell wall division. And then later on, what happens after this stage? So this is the 2-celled stage. Then you have the 4-cell stage, and then you have this 8-celled stage, or you can say 8-nucleate stage. This is 4-nucleate stage, this is 2-nucleate stage, and then after this there is a formation of the cell wall, and then finally the antipodals, they are formed at the chalazal end. Egg apparatus is formed at the micropylar end, and in the center is present but 2 polar nuclei. Clear? So the functional megaspore, this undergoes 3 mitotic divisions. So after every mitotic divisions, 2 cells are formed. Clear? So there is one functional megaspore. This gives rise to 2 cells. So this is the first mitotic division. This is the second mitotic division. This is the third mitotic divisions. Okay. Although all these are mitotic division. Clear? First mitotic division, second mitotic division, and this is the third mitotic division. After 3 mitotic division, 8 cell stage is achieved. Now what will happen? After this, cell wall formation will occur. Clear? So the first mitotic division produces 2 nuclei, one larger and one smaller, establishing the basic organization of the embryo sac. So they are talking about this 2-celled stage. Now, last is the formation of the embryo sac structure. So the larger nucleus migrates to the micropylar end and becomes the egg cell, which is also known as the female gamete. The 2 nuclei located in the central cell fuse to form the diploid nucleus called the secondary nucleus or the central cell. The remaining nuclei are positioned at the chalazal end and contribute to the formation of the antipodal cells. Okay. Clear? So antipodal cells are formed at the chalazal end, egg apparatus is formed at the micropylar end, and in the center 2 polar nuclei are present. Clear? And later on, in the mature embryo sac, the 2 polar nuclei fuse to form a diploid nucleus, which is also known as the secondary nucleus. Clear? And these polar nuclei, these 2 polar nuclei, these are located in the central cell. Clear? Now we know that the pollen tube, okay, it travels through the style and then it enters into the embryo sac through one of the synergid, and the filiform apparatus helps to guide the pollen tube entry into the embryo sac. Now what we are talking about the events that are happening after the pollen tube enter into the embryo sac through the synergids. So the pollen tubes, they release the 2 male gametes into the cytoplasm of the synergids. So where are 2 male gametes released? These are released in the or within the synergids. Now one of the male gamete moves towards the egg cell and fuses with the nucleus, and this is known as the syngamy, and this results in the formation of the diploid cell known as the zygote. Clear? So what is happening here? One of the male gamete. So there are 2 male gametes. We are talking about the first male gamete here. first male gamete, it fuses with the egg cell. Okay, so both are haploid. Male gamete is also haploid. Egg cell is also haploid. All the cells of the embryo sacs are haploid. Clear? So this leads to the formation of the zygote. So zygote is a diploid unicellular structure. So zygote will be 2n, means it is diploid. It contains 2 sets of chromosome. Now what is zygote? Zygote is a unicellular, means single celled. It is unicellular, but it is diploid structure. Diploid structure. Clear? Now this is known as the syngamy. This is known as syngamy. Syn means fusion. The fusion of the 2 gametes is happening here. Okay, this is known as fertilization also. Fertilization also is the union of the 2 gametes. Okay, here we are calling it as syngamy. The fusion of the first male gamete with the egg cell, this is known as syngamy. And this results to the formation of the zygote, which is a diploid structure. Clear? Now the other male gamete, what is? Let's talk about the other male gamete. So the other male gamete moves towards the 2 polar nuclei, which is located in the central cell, and fuses with them to produce a triploid primary endosperm nucleus. So let's PEN. Okay. So let's talk about the second male gamete. Second male gamete is also haploid. It fuses with what? It fuses with the 2 polar nuclei. So these 2 polar nuclei, okay, they fuse to form the secondary nucleus we have seen. Okay, these 2 polar nuclei fuse to form secondary nucleus. Okay, the secondary nucleus will be then diploid. So we can say the second male gamete fuses with the secondary nucleus, or the male gamete fuses with the 2 polar nuclei, resulting in the formation of what? What will be formed? A triploid structure will be formed, means 3n, which is known as the primary. It is known as the primary endosperm nucleus. Okay, this is known as the primary endosperm nucleus, and it is a triploid structure. So this process, this is known as what? This is known as triple fusion. Okay, this is known as triple fusion, and collectively both syngamy as well as triple fusion, this is known as double fertilization. Clear? So what is happening in the double fertilization? During double fertilization, what is happening? Syngamy, which is the fusion of the one of the male gamete with the egg, leading to the formation of the zygote. And second one is the triple fusion, fusion between 3 cells. The second male gamete this fuses with the 2 polar nuclei, or you can say the secondary nucleus. So this is known as double fertilization, and this is very important. Lot of questions can come from this topic. Okay, clear? Now this is an image. So in this image you can see what is happening after the fertilization. Clear? So this image is showing you the embryo sac, condition of the embryo sac after fertilization. So here fertilization has taken place. So after fertilization, what happens? A zygote should develop. So this is the zygote, which is a diploid structure. This is formed by the fusion of the male gamete and the egg cell. Now second is the formation of primary endosperm nucleus, which should be triploid. And this is formed because of the triple fusion. And then this is the primary endosperm cell. Okay, so central cell get transformed into the primary endosperm cell, and this primary endosperm cell, this consists of the primary endosperm nucleus, which is triploid. And what other things are happening? The antipodals degenerate. So antipodals, these are present at the chalazal end. And synergies, these are present at the micropilar end. So the antipodals also degenerate and synergids also degenerate after the fertilization, and egg cell, okay, it fuses with the male gamete and it forms the zygote. Okay, clear? So what things are happening here? Okay, so you can write after fertilization what is happening. First of all, what is happening? Zygote is formed. Okay, zygote is a diploid structure. Then a primary endosperm nucleus or the primary endosperm cell. Primary endosperm cell is formed, which consists of the primary endosperm nucleus. This consists of the primary endosperm nucleus, which is triploid. This is present. And then what happens? Synergids will degenerate. Okay, synergids degenerate and antipodals also degenerate. Clear? So these are the events that are happening after the fertilization. So the embryo sac will contain the zygote. So zygote will later on, okay, what it will form? It will form the embryo. Okay, and the primary endosperm, this cell, this will form the endosperm. Clear? So you can write here the zygote which is formed. Okay, so we can write here only the zygote, it will form the embryo, and embryo will form the new individual, and the primary endosperm cell, it will form the diploid structure known as the endosperm. So the endosperm will be triploid. Okay, embryo will be diploid. Clear? And what is the function of the endosperm? This endosperm, this provides nourishment to the what? It provides nourishment to the growing embryo. Clear? Now let's move on. Now, we have already talked about the triple fusion also. Triple fusion involves the fusion of the 3 haploid nuclei. So all the cells in the embryo sac are haploid, as well as the male and the female gamete, these are also haploid. Now, since the 2 types of fusion is taking place, one syngamy and another one is a triple fusion, and these 2 fusions are taking place in the embryo sac, this phenomena is termed as double fertilization, and this double fertilization is unique to the flowering plants. Now, what are flowering plants? These are the angiosperms. Flowering plants are the angiosperms. Clear? Now, the central cell after the triple fusion becomes the primary endosperm cell. So, primary endosperm cell is formed from the central cell. Okay, central cell consisted of what? Central cell consisted of the two polar nuclei. Clear? So primary endosperm cell is formed from the central cell and it develops into the endosperm, while the zygote develops into the embryo. Clear? So you can write here like central cell, which before fertilization it contains the two polar nuclei. Central cell after fertilization will develop into the primary endosperm cell. Okay, PEC. And this primary endosperm cell will form the endosperm. Endosperm is a triploid structure and it gives nourishment to the growing embryo and the zygote. Zygote will form the embryo and embryo will further differentiate. Embryo will further differentiate and it will form the new individual. Okay, or you can say new plant. Clear? So this plant will also be diploid because embryo is diploid and zygote is also diploid. Clear? So what we have studied, we can summarize that. So let's summarize this entire topic. First, we have studied the development of the embryo sac. Okay, development of the embryo sac. So briefly, how can you write? First is the megaspore mother cell formation. Megaspore mother cell is formed. Okay, so this is a diploid cell and it undergoes meiosis. Meiosis is what? Meiosis is the reductional division. And after meiosis, 4 cells are formed. Okay, so meiosis, you know, it has 2 phases: meiosis 1 and then meiosis 2. So after meiosis 1, 2 cells are formed. Okay, and after meiosis 2, 4 cells are formed. So meiosis 2 is same as mitosis. Clear? So after meiosis, a megaspore tetrad is formed. Okay, means it consists of 4 cells. Now, out of these, 3 will degenerate. 3 degenerate and only 1 is functional. So the functional megaspore which is haploid now, okay, this will undergo 3 mitotic divisions. To form 8 nucleate and 7-celled embryo sac. Alright, so you have to remember the mature embryo sac is 8 nucleate. There are 8 nucleus. Okay, and it is 7-celled because the 2 polar nuclei, they fuse together to form the secondary nucleus. So it is 8 nucleate but 7-celled. Alright, clear? Now we have studied about the we have also studied about the double fertilization. So in double fertilization, what happens? Syngamy as well as triple fusion is happening. Clear? So I hope you enjoyed this lecture. Thank you very much for listening and we'll continue with rest of the topics in the next session. Thank you very much.